Okay, well, thank you for joining us today, everyone, and a very warm welcome to you. Lovely to see a good number joining us at the end of the busy year, and really pleased to welcome Florian Schiestl. Thank you so much for joining us, Florian, all the way from Vancouver. Although Florian is based at the University of Zurich, he's currently on sabbatical in Vancouver. So it's pretty early in the morning for him there. So just before we get going, if I could just remind anyone who wants SACNAS points, if you could please put your name in the chat and indicate that you need them. Please put your full name so that Elizabeth can assist you. And then I'm going to hand over to Sandy Steenhuisen, who's actually out in the field. So let's see if Sandy can join us to introduce Florian.
Thanks, Glynis. Here. Can you hear me?
Yes, we can. Go ahead.
Awesome. Great. Yeah, good morning to Florian and evening to everybody else in South Africa. And yeah, we have the privilege of inviting or having Florian with us today to speak about his work in biotic interactions and plant evolution and the role of biotic interactions and plant evolution and divergence. And yeah, just a bit of background. Florian is a professor in ecology at the University of Zurich. And we must have met about 15 years ago in Steve Johnson's lab in Pietermaritzburg at UKZN when I was still a postgraduate student. And then we've met at a couple of conferences then. So it's great to see him again, even virtually. But yeah, he's a leading researcher in ecological and evolutionary questions on plant pollinator interactions, specifically on pollinator-driven speciation, deceptive pollination systems, mostly including deceptive orchids, where they are tricking pollinators into visiting their flowers because they resemble their mates. Yeah, Florian completed his master's and doctorate degrees in evolutionary biology at the University of Vienna and teaches mostly plant diversity and species interactions at the University of Zurich. He's been on the editorial board of Ecologia for 6 years and is currently on the editorial boards for Communicative and Integrative Biology and Frontiers of Ecology and Evolution. And yeah, besides supervising numerous master's, PhD candidates and racking up an enormous over 9,000 citations of publications, he's published in Science, Nature, Ecology Letters, Evolution, and other really highly acclaimed scientific journals. So we're really excited to hear what Florian is going to share with us today. And yeah, without further ado, I'd like to welcome Florian to start with this presentation.
Okay, thanks a lot, Sandy, for a very nice introduction. And yeah, I wish you all a good, nice spring evening. Here it's an autumn morning with a lot of rain and cold weather, so I bet you have a better condition in the Southern Hemisphere. My talk about the role of biotic interaction in plant evolution and divergence starts with a bit of an emotional introduction with the first slide, showing just some of the amazing beauty that comes with it, and namely that's the diversity of flowers. And I would like to make the argument that a lot of us are coming into biology because we just have this affection to the object, namely the diversity of different organisms. In this case, it's plants, or especially the flowers. And so these Australian terrestrial orchids, of course, very famous for their floral diversity. And you have similar examples in the genus Daisa, for example, in South Africa. I mean, South Africa, anyways, very, very famous for its diversity in plants and in flowers, especially so. And very, very many good examples of what I'm talking about actually come from South Africa, from the research that has been done there. But I worked mostly in Europe, but also in Australia during my postdoc. So I'm showing this Australian example here. And it is, of course, an old idea that this floral diversity is a consequence of plants adapting to different pollen vectors, that is, biotic pollinators like insects or birds or bats. And this, as a consequence, leads to the different floral morphology, floral fragrance, floral color, and so on. So the signals and the shape of the flowers that ensure efficient pollen transfer. That's the sexual function of the plant. But I'd like to make the point here that this process or this phenomenon is understood well in terms of pattern, but not so much in terms of process. So we don't really know very well how this process starts, what are the main driving factors, because often you have covariance. For example, plants with different pollinators often also grow on different soil types, and we don't really know actually what is the first primary drive of divergence. Is it the soil type, and then secondarily comes the adaptation to different pollinators, or the way around? So these kind of things are not very well known because we simply don't have much of an experimental approach into this phenomenon, because it's considered a long-term evolutionary phenomenon. And then also plant groups like orchids don't lend themselves very well for experimental approach, because they are very difficult to grow from seeds. They have mycorrhiza that's needed for germination, and they have very long generation time. So it's just a difficult system to work with. And you can mostly just study it in the field. And I've worked a lot with orchids, but about 8 years ago, I made a strategic decision to switch to a new system, and that's the genus Brassica, simply because it allows much more rigorous experimental approach and to address a whole new set of questions. And that's what I would like to talk about in the second part of my talk. But let's go on. Let's see. How can I switch this slide? Okay, so the pattern of pollinator-driven divergence or speciation, I would argue, is just a special case of divergence, is known from, for example, phylogenetic studies that often found that cladogenesis, or the formation of new clades, is often associated with the switch to a different pollinator or pollinator group. And here we have examples from the Gesneriaceae from Southern America. And then here, a North American example in the genus Aquilegia, we have similar phenomenon. But as I've said before, there are other factors that play a role. Covariance with abiotic factors, for example, climate or soil type, is very commonly found. And thus we don't really know what drives the onset of this process. Just a few conceptual outlines about how this process works, how we think it works. There are basically two different possibilities. One is a sort of a classic allopatric scenario of divergence. And the second one is a scenario that can also work in sympatry. So here we have the association between selection and then and then evolutionary trait change in the plants. The idea how the adaptation to different pollinators leads to the evolution of different flower types. So, for example, this is just a hypothetical example here, the evolution of long floral spurs here, or species with short spurs and long spurs here, driven by the adaptation to a long proboscis pollinator. So this long proboscis pollinator here will drive directional selection in the direction of longer spurs, because more fitness is associated with longer spurs. And this is not the case for the species that has a short proboscis pollinator. This directional selection leads to anagenetic change. So that is the evolution along the evolutionary line and the increase of this floral spur. And then in the end, we have species with different floral traits, in this case, different spur lengths. And the side effect of this can be reproductive isolation between the two species, simply because they don't share pollinators anymore. They are divergent enough to have separated in the pollinator use, and therefore reproductive isolation is a byproduct here. And this form of reproductive isolation has been termed floral isolation, because it is the flower with the interaction, or in the interaction with the pollinator, that mediates this kind of isolation. And the person I'd like to give credit here to is Vern Grant, who worked a lot on this concept, and yeah, for the first time worked on it in a very deeply way, I would say. And later it was shown that in several more specialized pollination systems, this is a very important and very strong reproductive barrier. It can be very strong because it is a barrier that acts very early in the sequence of different barriers. So for example, post-zygotic barriers act relatively late, act after the formation of a zygote. And floral isolation acts very early, before the pollen is actually transferred between the different species. So it can be very effective. It's also a very parsimonious isolation, because pollen is not wasted onto incompatible mating partners. Yeah, and so in these systems, the flower mediates both reproductive success and also reproductive isolation. And these dual functions of the flowers led to their naming as so-called magic traits in speciation. And just briefly explain these examples here. This is the genus Mimulus, where we have famously bee-pollinated and hummingbird-pollinated species. Then Petunia here, you also have the bee-pollinated forms, and then hawk moth-pollinated species. Here the genetics have been worked out very well in the lab of Chris Kuhlemeyer in Bern. And these are sexually deceptive orchids in Europe, the genus Ophrys, which have a special form of reproductive isolation, which works on a species-specific basis. Because here pollination is so specific that even individual plant species have just one pollinator species, and here this barrier is highly specific and is also very effective. And we have worked on this system a lot. Okay, so how can this lead to diversification? These are the two scenarios I'd like to introduce. First, the classical allopathic scenario. Here we have a directional selection in different forms here, which is called divergent selection. For example, one-time selection for longer spur and one-time selection for shorter spur, which is driven by a geographic mosaic of different pollinators. This geographic mosaic by Vern Grant called this the pollinator climate. So it's kind of the biotic mosaic of different factors that drive different forms of selection that eventually lead to diversification, simply because in different regions, plants adapt to different pollinator. This is an example from South Africa. This is the distribution of long-tongue flies on the landscape of South Africa. Classic example of this pollinator climate here, which then leads to allopathic diversification. And this is the asympatric variant of this concept here. And this works not with divergent selection. Divergent selection, by definition, is selection in different population. Here we have disruptive selection. Disruptive selection works within one population. And here we have selection for the extremes. So for example, in spur lengths, it would be selection on either very short or either very long spurs. And this is thought to be driven by negative density-dependent selection. Simply assuming that if plants that use one resource of pollinator become too common, they will compete for this resource. For example, if they use short proboscis flies a lot, at some point there will be a lot of competition for short proboscis flies. And then any mutant that uses long proboscis flies by the assumption of a longer spur has an immediate advantage, because it basically uses a pollinator's resource that there is no competition on. So this is this idea of negative density dependent selection in a very simplified form, of course. And this again can lead to diversification, in this case to sympathetic diversification through the adaptation of different sympathetic pollinator species. Okay, so far for pollinators, but I would also like to talk a little bit about the effect of herbivores. Herbivores are animals that consume parts of the plants, and herbivory is a very common phenomenon in nature. Of course, almost any plants have any forms or any kinds of herbivores. It can be herbivores. In Africa, of course, the big megafauna herbivores are very prominent. But also insects, of course, like butterflies, for example, lay their eggs on plants, and then the caterpillars feed on the plants. This is, of course, very important and common as well. And herbivory has a clear impact on plant fitness. It can have direct effects. It reduces resources, so it reduces some tissue, or even directly the ovules or the seeds or the flowers. So these are direct effects of herbivory. But herbivory can also have indirect effects, because herbivory often triggers a form of phenotypic plasticity, and that is the upregulation of defenses. So plants respond to herbivory by producing more toxic compounds. And this has been shown in response to insects, but also in response to herbivory, to vertebrate herbivory. So it is again a very common phenomenon. And this upregulation of defense has an effect of pollinators too. It has been shown that some of these defense compounds travel into the rewards, for example, into the nectar. And then, because the nectar becomes slightly toxic, it impacts the visitation behavior of the pollinators. And this can have indirect effects then, obviously, because pollinator behavior is a very important selective force for flowers. So we have an indirect effect on floral evolution. This is something I'd like to talk about later in the talk. Okay, so these are my main questions for the talk and my two study systems that I'd like to introduce to you later. We first talk about divergence and reproductive isolation in a sympathetic scenario. And here I'd like to use the ORCID system that we've been working on a lot in the more longer past, the genus Ophrys here, the floral mimic with a very specific pollination system. And then the second is the allopathic divergence with different pollinators and herbivores, more a classical system. And here, this of course is a less controversial scenario. It's relatively well understood, I would say. But the main novelty of my research here is that for the first time, we have given an experimental proof of the process, how this works in the very onset when plants really start to adapt to a new pollinator. And for this, we have used experimental evolution with Brassica rapa. Brassica rapa makes this possible because we have genotypes available with a very short generation time that we can really let evolve over about 10 generations and then see the evolutionary change that happened during this time. Okay, but first start with the ORCID system, the genus Ophrys here. And this is just a little general introduction into the phenomenon of floral mimicry. Floral mimicry works with flowers imitating an item of interest for the pollinator. And this item is not always food, but it can be different kinds of things that have a very high ecological relevance for the pollinator. And therefore, the pollinator is strongly interested in this item. So this item can be a sexual partner. Here, this is the case of sexual mimicry. And the pollinators are males, only male insects. Of course, their interest is to mate with a female. And that's what they do on the flowers. They try to mate with those flowers because the flowers imitate the mating signals. The item can be food here. This is a food mimic, South African Aldaisa, that mimics Scabiosa and has a very specialized pollinator. So the Scabiosa has the specialized butterfly pollinator that will also visit this Daisa, and but the Daisa has no food, so it's a food deceptive system. And then the other items that I show here are oviposition substrates. Here, this is a kind of dung mimicry. The pollinators are dung beetles here that normally lay their eggs into dung. This is, of course, very important for the biology, and the beetles have a very hardwired preference for dung volatiles. And they will visit these flowers because they think it's an oviposition substrate. The same is true for the Stapelia here. This is a carrion mimic. These plants are not the ones that you would put into your living room because they stink like hell, like a decaying piece of meat. And the pollinators are blowflies here that again try to lay their eggs. Sometimes they even lay their eggs there, so it's a very effective mimicry. So if you want to know more about floral mimicry, I can recommend this book here written by Steve Johnson and myself, which is, I think, the only summary coverage of this concept, and I think it's an enjoyable read. Now I will be talking about sexual mimicry, and I'd like to also give you one example from the South African flora of this. We have a few, not many examples, but there are a few species of the genus Daisa that have evolved sexual mimicry. And this is the most recent discovery. I should say that sexual mimicry is being discovered all around the world now as we're speaking. So we have new discoveries from Australia, from South America. South America is very little investigated, and likely there will be many, many examples popping up in the next decades. I hope so, at least. Many plants look like they would be sexual mimics, but they have just not been worked on. This is the Disa forficaria, which is an excessively rare plant, has been discovered in the 19th century. Then it was flowering in 1966. It's all described in this paper here by Cohen et al. Steve Johnson is the last author. And then it was thought to be extinct after 1966. It has not been found anymore. But then a flowering individual was found in 2018, and that study is based on the single individual. And these authors have used this individual to work out the attractive chemistry of this plant. Sexual mimicry primarily works with chemistry. So it is the floral odor that attracts the pollinator. And in the next year, this plant was again not flowering anymore. And they could not find it anymore, but they did bioassays with the attractive compound. And they successfully attracted the pollinator, which is a longhorn beetle. And some of the pollinators that came in these bioassays were actually carrying pollinaria. And the pollinaria were sequenced, and it was confirmed it is from this Daisa species. So, but nevertheless, they could not find the flowering plants, but they knew it must be in the area. So I think it's an exciting example of how the pollinator can be used to find hidden individuals of a rare orchid. So I really recommend this paper. It's a very enjoyable read. Okay, but let's go back to Europe. This is the system we have been working with. It's a small monophyletic subclade of the genus Ophrys, the Ophrys insectivora clade, which only has 3 species. It's a monophyletic clade that makes it very attractive. So we know they have a common ancestor. And it consists of 3 species with vastly different distribution in Europe. The classic Ophrys insectivora that was described by Linné. So it's a well-known species in Europe. It occurs up to southern Sweden, so it's widely distributed in Europe. And then we have 2 local endemics in this clade, Ophrys aimonini and Ophrys subinsectivora, that have different pollinators, all very specific. So Ophrys insectivora has a digger wasp as pollinator. Ophrys aimonini has a solitary bee, and Ophrys subinsectivora has a sawfly. This is also very unusual. It's the only Ophrys species with a sawfly as pollinator. And here we have the distribution. Ophrys aimonini is a local endemic in southern France, and Ophrys subinsectivora a local endemic of northern Spain. Okay, and we worked in southern France with this, not only because southern France is a beautiful area with crusaders' castles and a nice cheese with strong olfactory signals. The interesting local specialities include the crop of the ducks. Other endemic orchids, it's a wonderful area, the Aveyron it's called. But it was also the more amenable system for us. We found the bees in the wild. We could do bioassays there, so it was the clear choice for us to work with. So we chose Ophrys aimonini and Ophrys insectivora, which grows highly sympatrically there in the area. So usually within meters, you find the species flowering together. So this is a clear case of sympatry, broad sympatry. And then this is the student who worked on this, Daniel Gervasi. Daniel did experiments to check the pollen flow between the species, whether there was pollen flow between the species, which would lead to hybridization of the 2 because there is no genetic barrier. That's also, he checked also that. He did crosses, artificial crosses between them, and they happily produce seeds. There's no post-pollination barrier in these species. But the floral isolation is very strong. So by staining pollinaria here, and then monitoring the flow of stained macula between the species, Dani showed that there, at least in our observation period, there was no interspecific pollen flow between the species, only intraspecific. So floral isolation, the conclusion was, is very strong in this system. And this is the same that we have shown. We've done these kind of experiments with a handful of systems. And we have found that floral isolation is very, very strong in these systems. So it is a very strong reproductive barrier in the genus Ophrys. And then we went on to work out the attractive chemistry here in this system. We've used electrophysiological recordings for that. So this is a method that has been developed in the 1960s to identify insect sex pheromones. And it enables the ability to take the nose, that is the antenna of an insect, and then blow the compounds that elute from a gas chromatograph over the antenna and record the electrical signals that are created in the antenna when something is smelled by the bee or by the insect. And these are the deflations here in this EID curve. You see here, the compounds that cause such a deflation are the so-called active, biologically active compound, if you want. And these are the ones that can be smelled by the pollinator. And we identified these compounds, and the compounds, these active compounds were alkenes, so unsaturated straight chain hydrocarbons, and long chain wax esters. And as you can see here from this graph, these 4 active compounds are also the ones that are significantly different in amount between the 2 orchid species, which already suggests that they are probably the compounds that cause the different pollinator attraction. But we've tested for this with synthetic copies of the compounds. And here I'd like to give credit to my long-term chemical collaborator in the Ophrys system. This is Professor Witko Franke, who very sadly passed away about a year ago. And Witko was a very dedicated researcher. He was already retired when we did this study, but he still had a little lab at the University of Hamburg. He had no students anymore. So when I called him and said, like, I need these compounds for bioassays, he went to the lab himself and cooked the compounds for me so I could do the bioassays in the field. I think this is just truly extraordinary, and he deserves, definitely deserves a place in the Hall of Fame of chemical ecology. And I sincerely thank him for his contribution, important contribution for my career. So these compounds we tested in the field, and how we did this, we basically added these compounds to the orchid flowers that don't have them, namely the flowers of Ophrys insectivora, and we offered the flowers, then these manipulated flowers, to the bees that normally pollinate Ophrys imonini. And we saw that indeed this manipulation of the flowers led to the attraction of the bees. And here you see a photo of this. This is a photo of a bee attempting to copulate on the flowers of insectivora. This is very strange because you would never ever see this with normal insectivora flowers. Here we have the insectivora control without manipulation. The bees are not very interested in these flowers, but as soon as you add these compounds, you see that the bees start to pounce and attempt to copulate. So these 4 compounds are clearly the key chemical signals that led to the switch of pollinators from digger wasp to bee pollination in this system. So we have strong floral isolation. We have the traits that trigger the pollinator switch. And one more thing we need for this symbiotic speciation scenario, and that is negative density dependent selection. That would lead to disruptive selection in the population. And we have also shown this, at least in one of the species, namely in Ophrys insectivora, across several populations. We've done about 20 populations that have sympathetically the 2 species. And indeed, there was overall a negative association between the relative fruit set and the density defined as the number of conspecifics within a 2-meter radius in the field. You see, this is a negative association. So here, this is shown that there is negative density dependent selection, and this is at least a smoking gun for disruptive selection in the populations. There was a trend for the same thing in Ophrys imonini, but it was not significant in this case. So we would argue that here, because there is negative density dependent selection in very dense population, mutants that use the bee, that assume the new scent bouquet, the scent cocktail, and use the bee as a pollinator, have an incident advantage, leading to the establishment of them, and also leading to strong floral isolation. So there will be little or no gene flow between these new incipient species and the ancestral species, and that can lead to sympatric speciation in this system. But of course, I happily admit that this is the study is not complete. Most importantly, because we have never shown that the pollinators here, the pollinators of insectivora, the digger wasps, would negatively respond to this new scent cocktail. And this, of course, would be a requirement. Otherwise, the isolation would only be one-sided. And there could still be gene flow through the one pollinator that would not be deterred by the bee attractive scent compounds. The problem is just we have never seen any of these digger wasps in the field, although we've studied the system for 4 years. These pollinators are just enigmatic and we have not found them. This is one of the classic examples that orchid stories are often intriguing, but leaky. So it's kind of a frustrating situation that it's very hard to construct a waterproof case, especially in speciation where you have so many different ecological factors that are important. And yeah, this is just the summary here. So in these specific pollination systems, we have very strong floral isolation. That's based on relatively simple traits, the 4 scent compounds. We have negative density dependent selection, so pollinator mediated speciation can be sympatric. So far so good, but as I said, it's a little frustrating. We cannot make this waterproof, and this was one of the reasons why I decided to switch to a new study system about 8 years ago, enabled by a fund from the European Research Council. I started to use experimental evolution with the genus Brassica, more specifically Brassica rapa. And you may think, okay, Brassica as compared to orchids is very boring, but it is a very fascinating system. It doesn't have much floral diversity. Brassicaceae are known for their relatively uniform flowers. There's not much floral radiation there. But on the other hand, Brassicas are very diverse in their growth form. Also, we have different cultivated forms. For example, the vegetables, the cabbage or cauliflower, what else? Broccoli, Brussels sprout. It's all botanically one species, namely Brassica oleracea, the vegetable Brassica. And then Brassica rapa has, for example, Napa cabbage, an edible cultivation. The two are parent to oilseed rape, which is a very important crop species, probably the most important crop species grown in Switzerland. So we have a lot of vegetative diversity. We also have a lot of variation in the scent. I've often smelled Brassica flowers. They can be very divergent, different smelling. I've picked up very strange smells in Italy, for example. Some Brassica smell like burnt plastic. Others have like a very sweet, deep sweet aroma. Others are more terpene, more like a pine terpene aroma. So it's a fascinating diversity that's hidden there and not so well investigated at all. Brassicas also have the advantage that many features are really well worked out. So it's a well-known system. For example, the defense system has been worked on by many people. It's the so-called mustard oil bomb. The system with glucosinolates and myrosinase enzymes that are stored in different compartments in the cell. And when the cell is crunched by a herbivore, the two come together, and the myrosinase split the glucosinolates into so-called isothiocyanates, which have a very pungent, strong, hot sensation. And actually, you all know this sensation because it's what we like about the flavor of mustard or horseradish, for example. So this is hot sensation. These are isothiocyanates, which are the classical defense compounds of Brassicaceae. This is wasabi, by the way, here, the Japanese horseradish. Then pollination in Brassicaceae is typically generalized. There are a few genera which have more specialized pollination, but it's more the exception than the rule. So quite different from the orchids. But this is also nice for experiments because we can test, we can use different pollinators and test divergent evolution with different pollinators. This the generalized pollination enables this. And the clue about this whole thing is we have these fast cycling genotypes available. And they were developed by Paul Wilson at Wisconsin University for research and teaching. And they are also a great tool for teaching. So I also use them in teaching a lot, actually. And they have this fantastic short generation time of about 7 weeks. So these plants are so fast. When you grow them, the seeds, you drop the seeds in the soil. 2 weeks later, they will flower. So it's really fast. And from seed to seed, it takes about 7 to 8 weeks, depending on the growth conditions. So in one semester, you can go through several generations, 2 or maybe even 3 generations, and you can do genetics. You can show basic genetics. You can also do pollination experiments in a class, for example. So it's a wonderful tool for teaching, but also for research. As I'd like to make the point here, because other than being fast cycling, these are fully fledged normal plants. They are outcrossing. They have genetic self-incompatibility. So they're fully outcrossing, which is a prerequisite for pollinator experiments, of course. They are attractive for pollinators. They have reward. And this is in opposite to Arabidopsis thaliana, for example, which is a sulfur, which is not very attractive to pollinator, and it's not useful for pollination research for that reason. Still, it has Brassica rapa has a small genome size. It's diploid. It is about 4 to 5 times the genome size of Arabidopsis, so it's also good for genomic approaches. And this is what I'd like to do in the future a lot more. And then we have interacting pollinators. And the nice thing here is we can buy them. We can buy Bombus terrestris. It's being produced by tomato growers. So we have a Swiss company. Its headquarters is in the Netherlands, actually. And they produce bumblebees, and we can buy the bumblebees all year round, which is wonderful. You can also buy syrphid flies. They are used for biological pest controls because their larvae feeds on aphids. And we breed Pieris as an herbivore that we also use as a biotic interactor in these experiments. This is the basic design of these experiments. We control for genotype by using full zip families that we create ourselves. It's very easy to do compared to orchids, where it's basically impossible to do. Then here we have the treatments, for example, different pollination treatments. And the treatments have replicates. It's nice to do replicates so you can show that the things are reproducible within one experiment. Replicate size can be 36. In the past we have 36. Now we actually use 50 plants, which is better for inbreeding because the replicates are carried through as independent lines in these experiments. And if the replicates are too small, there will be a lot of inbreeding, which is not nice to have. So we use now replicate size of 50. And this is how this experimental evolution works. We have this is a replicate here, then we do the pollination, for example, hand pollination for the control, or we release the bumblebee in a cage. Here we have the flight cage in a greenhouse, or the hoverflies, depending on what pollinators we want, or Pieris, we also use Pieris butterflies as pollinators. And the seeds that are produced from this pollination are carried on to produce the next generation. And each plant is allowed to contribute an amount of seeds that is proportional to its total seed production. Of course, we cannot take all the seeds because that would be too much. We cannot have the populations grow. So we reduce it to a proportional contribution of each plant according to its overall pollination success. And this, of course, allows for selection to transfer into evolutionary change because the plants that have been very successful contribute more seeds in the next generation. And that's the principle of evolutionary change driven by natural selection. And then, the genotyping the phenotyping and genotyping is done then in the last generation. We do one generation in between where we grow the plants without insects to just avoid any maternal effects, and we can cross the replicates to overcome inbreeding and so on. So in the second generation, after the end of the experiment, we do the phenotyping. We do a lot of phenotypic traits that we can do in our lab. We do floral signals, for example, floral scent. Here we have the setup for collection analysis of floral scent. This is UV reflections. The white part is the reflections. The dark part is the UV absorbance. This is the classical bull's eye pattern of UV that the brassica flowers have. This is floral morphology, herkogamy, that the plants have. So a separation of the sexual organs in the flower. We also measure that. That's undergoing evolutionary change, as you will see. And this is the first experiment that was also done by Dani. And Dani now, he tested the onset of this, the classical allopathic speciation scenario. Plants grow in different regions, face different pollinator communities, and therefore evolve in a divergent way. So he used different pollinators. He used bumblebees and hoverflies in brassica, and then hand pollination as a control. And this experiment, this was the first experiment that we did, and well, I had no idea whether this is going to work. So Dani was a pioneer in this, and I can only say I was blown away by the success of this experiment, and he got a nice paper out of it. So it was a wonderful effort, and it basically was the initiation for us to do much more of this kind of research, because it was such a powerful experimental approach. So he asked the question then, yeah, how do different pollinators lead to divergent trait evolution? How quick is this process? Can we see evolutionary change in the 9 generations that he did? And which traits evolve first here? And this experiment, of course, the nice thing here is there's no covariance. We only had difference in pollinators, so only the biotic factors varied, but the abiotic factors were just left identical. So we used this one greenhouse with the same climatic conditions, the same soil, the same watering. So all abiotic factors were identical. It's just the pollinators that varied. So this is a nice way to address what is the exact effect of only pollinators in driving divergent evolution in plants. Good. So this is an assessment of selection here, phenotypic selection that is, and how phenotypic selection differs between different pollinators. This is just a mean value of the beta selection gradient measured with bumblebees and hoverflies in the 3 replicates. And all these boxes show traits that were under significant selection, and the dark yellow boxes show when selection was significantly different. So this, I would argue, is divergent selection. And divergent selection was found for plant height and 3 scent compounds. And this is already the outcome of this experiment, a multivariate analysis of the plants at generation 11. And here we see the 3 replicates of the 3 treatment groups here: the hoverfly replicates, then the bumblebee replicates, and the hand pollination control. And you see there was divergent evolution. Traits differ in the 3 treatment groups. And what's the very nice thing about this is that the replicates actually seem to have evolved in all the same direction. And this is strong evidence that it was actually selection that caused this evolution and not drift. Because drift is a random process, and per definition, with the independent replicates, drift should have caused them to be all over the place. They may have been different, but they should not have evolved in the same direction. That's only what selection can do, because selection is not random. Selection should cause the same evolutionary direction in the treatment groups. Okay, so this is just how the plants looked like after this experiment, and I just keep showing this picture because when I saw this the first time, it just blew me away. I couldn't believe how different these plants were after just having different pollinators for a very short time, just 9 generations. And also a trait evolved that I did not expect to evolve. This is the plant size. So the plant with bumblebee pollination were much taller than the plants with hoverfly pollination. So just an individual picture how these plants look like. So there was a strong phenotypic change in these plants with just different pollinators. This is the plant height here over the generation. So from generation 1 to 11, see that there's variation. There's sort of mostly the hoverfly pollinated plants have sort of this downward bend, which we think is caused by inbreeding. There's a lot of inbreeding, especially in the hoverfly pollination plants, as we will see. And then in the end, we see that the bumblebee plants were the tallest of those. Other traits also evolved in an amazing way. Here, the scent-to-scent compounds, 2 examples. This is phenylacetaldehyde and anisaldehyde, 2 compounds that we know the bumblebees use. because they are often honest signals. So they are, the amount of these compounds is associated with the amount of reward. There was another PhD student, Anina Knauer, she has worked on that, and she showed that these are honest signals. And honest signals are used by pollinators, they are quickly learned and then preferred, which makes a lot of sense because bumblebees look for the most rewarding flowers. So they like these scent compounds, and therefore they positively select for them. And the consequence is a massive increase of these scent compounds in the bumblebee pollinated plants, but not in the hand pollinated plants. And in the hoverfly pollinated plants, this compound even decreased here significantly. So this was also showing that these are plants going into kind of a selfing syndrome. And then to show that these changes are actually adaptive, we did choice experiments with the pollinators. So we asked the pollinators, do you prefer the plants that have been pollinated for 9 generations by your species? And this answer was clearly a clear yes for the bumblebees. They clearly preferred the bumblebee pollinated flowers, but it was a no or a don't care for the hoverflies. So the hoverflies had no preference for either bumblebee or hoverfly pollinated plants. So we can conclude that the changes driven by bumblebees are clearly adaptive. They caused the bumblebee pollinated plants to be more attractive to bumblebees. This is a nice proof for adaptive change in these plants. And this is what we found all over in all experiments. This was the pattern basically. If you let the plants be pollinated or evolve by bumblebee pollination, they will in the end be more attractive to bumblebees. Now, what happened in hoverflies? So trait changes were not adaptive, didn't cause more attractiveness to hoverflies. That's what I've said. But there was another very prominent change here, namely the plants pollinated by hoverflies evolved a lot more spontaneous selfing. Here you see the amount of spontaneous selfing in first generation plants. So I've said that the plants are genetically self-incompatible, which is true, but the system is leaky. There's a bit of selfing is always there, very, very few. And this does not change in control and bumblebee plants, but it changes a lot in hoverfly plants. So they increase the spontaneous selfing to a massive degree. And the explanation is relatively simple. It's just the hoverflies are basically lousy pollinators. But this kind of hoverflies, I should say, they transfer a lot less pollen in each visit compared to bumblebees. And therefore, plants are under strong pollen limitation, and strong pollen limitation selects for selfing. This is the classical reproductive assurance scenario, a major explanation for the evolution of selfing. And it's nice here that you could have showed this, that selfing can evolve with different pollinators that have different degrees of pollination efficiency. So from outcrossing, mixed mating evolved here, so a mix of outcrossing and selfing driven by hoverfly pollination. And this is a little bit an outlook into the future. This is a pilot study that I've done with Dr. Lea Frachon. And this is about the genomics of this. Of course, we're also interested in what genomic changes were caused by these different pollinators within the 9 generations. And to do so, nowadays, it's not so complicated anymore. We can sequence the whole genome, and we can look for single nucleotide polymorphisms, SNP markers. And in this study, we have just used about 4,000 SNP markers. Nowadays, it's standard to use about 100,000 SNP markers. When I started microsatellite work in about 20 years ago, we worked with seven loci of microsatellites in office. Nowadays it's changed massively. We have a lot more resolution in this. And this study basically showed that we see a very nice pattern of genomic differentiation. We see that, for example, if you look at the SNPs under selection, we find a lot of SNPs that are under selection. And if you look at the divergence of the groups by using only those loci under selection, we see that the bumblebee pollinated plants cluster very nicely together here. And they're very distinct from generation 1 plants here, A and B1, and also from the control plants, which segregate in different directions here. The drift is much more important probably here, but here nice pattern confirms that selection most likely drives this evolutionary change also on the genomic level. Okay, so far for different pollinators, I think this was very convincing. But now we've, in the next experiment, encouraged by the first success of the first, we've engaged in another one. This was another student here, Sergio Ramos Castro, worked on this experiment. Here we added herbivory to ask the question, how does herbivory impact on pollinator-driven evolution in an interactive way? And Sergio did these treatments here. He added herbivory by adding larvae of Pieris brassicaceae, which is a specialist herbivore on Brassicaceae, on the leaves of these plants. So he added on the leaves the herbivores, and then he did the same thing. He had hand pollination and bumblebee pollination, no hoverflies though. But he looked what are the additional effects of this herbivory on plant evolution. And this was also a big successful experiment here, published in Science 2 years ago, so it was a great success. So he asked the question, how does leaf herbivory impact floral evolution? And how does the interaction between bee pollination and herbivory impact floral evolution? And this interaction proved to be very strong and interesting. Now, this is the multivariate analysis of his experiment. Here we see hand pollination plants and hand pollination with herbivory. It's already interesting to see that just the addition of herbivory to hand pollination did not cause much evolutionary change. Here these groups cluster nicely together. But then bee pollination here is different, and bee pollination with herbivory is again different here. And the interesting insight here is that the addition of herbivory to bee pollination changes the evolutionary trajectory of bee pollination a lot, quite significantly so. These are the attractiveness tests here, the choice tests for the 4 groups done by Sergio here. And again, very nicely, we see that only bumblebee pollinated plants are by far the most attractive. The hand pollinated groups are not nearly as attractive. But then the bee pollination with herbivory group was significantly less attractive than the bee pollination group, which shows that these plants have to compromise. They are not able to evolve their maximum attractiveness for the bees as they are able to do without herbivory. So herbivory causes a sort of a trade-off in attractiveness and defense. And we think we know what this trade-off is, but let me just show you the picture also for self-pollination, because another interesting insight was that herbivory speeds up the evolution of self-pollination once again. So in bee pollination with herbivory, we saw that self-compatibility was increased, and also spontaneous selfing was significantly increased with herbivory. So the interaction between bee pollination and herbivory causes increase of self-compatibility and spontaneous selfing in these plants. And let me just say one more word to this. The mechanism behind this, we also looked into this, and we think we know what it is. It's actually, we found that the nectar in these plants, when herbivory is present, has a high concentration of glucosinolates. And this leads to a shorter visitation time of the bumblebees on the flowers, probably simply because they don't like the nectar, taste like wasabi. And this shorter visitation time leads to lower attractiveness of these plants, but also to an increased pollen limitation of these plants, and therefore selection for increased selfing. So we think that the interaction here causes this in this way, in this classical way, that the defense travels into the reward, changing pollinator behavior as an indirect effect of herbivory. Okay, right. Just a few words about another experiment that we did, and this is more like sort of giving in that, of course, what we do in the greenhouse is very simplified compared to the complexity of interactions that we see out in nature. This is very clear, and we know about that, and we appreciate this diversity. And this is just one study here that I'd like to show that it takes advantage of this diversity by looking at how selection change when interaction become even more complex. What happens if you add one more additional player in the system here? And this study was done by Nina Knauer. She was a PhD, also a PhD student, finished 2 years ago. She did great work as well. And she studied in the Valais here, beautiful Swiss mountain. Here you see in this valley, she did field work with another Brassicaceae, the buckler mustard, Viscotella levigata. And on these plants, she found that there are often crab spiders lurking. And the crab spiders are an interesting interactor that we don't know much about them. We don't know much about what they do, and we don't know much about the fitness effect. And the intuitive expectation is, of course, okay, these crab spiders, they catch pollinators, kill pollinators, as you can see on this picture. They should have a negative impact on the fitness of the plant. Okay, makes sense. But Anina really took this under scrutiny and asked, what is the actual effect? What do these crab spiders really do? Okay, yeah, here we go. This is the system here. And what she did, she studied, first of all, she found out in a sort of an explorative experimental system or experimental approach, she found that the crab spiders use the floral odor to find the flowers to sit on. And this was quite spectacular and fantastic, because I think there was only one study that showed this before, but they did not actually dissect the odors. And Anina could show that the crab spiders use only one scent compound from the bouquet to find the plants. And this scent compound is beta-oxymene, which is a monoterpenoid. And interestingly, it's also a scent compound that is also used by the bee pollinators to find the flowers. And this, of course, makes a lot of sense. A predator should use the same signals that the prey uses to be successful. Because, yeah, if you see the place that is attractive for your prey, you will more be likely to catch the prey. And Anina was then interested in how this behavior of the spiders to follow this scent compound impacts selection on the scent. And she measured selection in the field by calculating, with multiple regression, calculating the beta, the selection gradient. And she found there's positive selection for beta-oxymene when there is only bees. But when the spiders are present on the flowers, this positive selection is gone. So this is yet another example for how an additional interactor can change selection and likely downstream evolution in this scent compound. By the way, the mechanism here is not that the spiders typically catch and kill the bees, but actually the bees they see the legs of the spider. And this has been worked out in another study. And they avoid the flowers where the crab spiders sit, so they can avoid being eaten by the crab spiders. Now, Anita found that crab spiders not only eat pollinators, but they clean up the flowers of any living being that's on there. So they're very greedy and generalist predators, and they also feed on caterpillars that live in the flowers. These caterpillars, herbivores or florivores, are of course very strong antagonists for the plants because they feed on ovules and seeds, and they have a very strong negative fitness impact. So Anita asked the question, so now it's getting complicated. What's the actual net benefit or net effect of these spiders? And the nice thing is you can study these spiders in the field with an experimental approach, because you can collect the spiders and then you can put them on the flower and they will just happily sit on these flowers. They will not move. So Anina experimentally manipulated flowers with and without spiders and with and without herbivores. So she added herbivores to the flowers, she added spiders in a full factorial design in the field, a beautiful experiment. And she looked at the fitness impact of those interactors. And first of all, she showed here that caterpillars with spiders were strongly reduced. Damage on the flowers was strongly reduced. And here's in terms of seed set, control plants, then plants with florivores have strongly reduced or highly reduced seed set. With spiders alone, also a slight reduction in seed set because pollinators get chased away. But then if you have crab spiders and florivores, you see that there is a net positive benefit because the negative effect of the florivores is being reduced with the spiders. So the answer here to this question is, that's a very nice answer. I think that there's a context-dependent effect of these spiders. When herbivore is present, it's a positive effect, and when no herbivore is present, it's a negative effect. And I think this is very commonly the case. I think this is a classical scenario that we have to pay more attention to. That interactive effects are context-dependent and depend on on the network of interactions that we find in the field. Okay, now, yeah, I'm already at the end. That's it. This is the summary of the findings. You've seen that different pollinators lead to rapid divergent evolution. Nice confirmation of the classical allopathic speciation scenario. Fluorescent and mating system evolved particularly quickly with different pollinators. So these interactions with different biotic agents are important for floral evolution. It's not just pollinators, but also herbivores and predators like crab spiders. They have important effects, interactive effects on floral evolution. But the end, maybe as a take-home message, I think we have clearly shown that biotic factors alone are a very strong evolutionary force for diversification in plants. And with these last words, I'd like to just give a very brief outlook into future experiments. We are now in the process of evaluating another experiment done by Thomas Dorey. And Thomas looked at the effect of soil. He used different soil types with experimental evolution, plus pollinators and herbivores, to look at the impact of the interactive effect of abiotic and biotic factors. And this is something that interests me a lot. So I'd like to work on this more in the future, because just again, going more natural, because in the field we have abiotic and biotic factors that vary, and how they impact the evolutionary trajectory of flowers. I think it's a very fascinating story. And this is the team that I'm working here in Vancouver on genomic approaches more. I'd like to learn more about this because I think it's a very fascinating evolutionary avenue, experimental avenue for studying evolution. Okay, so I've already given credit to some people that were involved. Maybe additionally here, Alice Palmer, she was a technical assistant, and she had a lot with lots of work phenotyping those plants. So she was very important help for us. And then, of course, our financing sources, the European Research Council and the Swiss National Science Funds. And thanks to you all for listening, and I'm happy to address any questions if there are any. Thanks very much.
Thank you so much, Florian. It was a fascinating talk and a lovely journey. We would love to always find these awesome study systems to work on. We're just racking our brains and trying to think of plants that have such rapid life cycles to do this with. But yeah, I'd like to open to the floor for any questions. Please feel free to write in the chat or just come on the microphone. You're very welcome. Perhaps I wanted to just start off by asking how often it's been shown that nectar is made bitter or toxic as a response to herbivores. If you found any other literature sources on that?
You mean if it's been shown in other systems, or?
Yes, yeah, and that the nectar's been made bitter and then has a consequence on pollinators. Yeah.
Yes. Yeah. I mean, one famous example is from South Africa. Steve Johnson's worked on in the aloes, genus aloe. I hope I'm pronouncing this correctly. The aloe, some aloes have dark and bitter nectar, which is not acceptable to bees. So bees smell this bitterness and they reject this nectar, but birds don't care. So this secondary compound changes the pollination system, turns this system to be bird pollinated. This, of course, is not induced defense, but there is another famous example is from Nicotiana. It's shown that if caterpillars attack Nicotiana, the amount of nicotine in the nectar changes, and this changes the behavior of the pollinating moth. The visits become shorter, so the moths shorten the visits and they visit more flowers. And this was actually interpreted in a way that this increases the male function of the flowers, so increases outcrossing rates, and it's actually a beneficial function of this increased nicotine concentration, which may very well be true. So it may also be true in Brassica, but we have not specifically targeted this. So they're different. I just want to say that there's actually different ways of interpreting this the secondary compounds in nectar. And I think it's a fascinating ongoing research area.
No, absolutely, yeah. Aryan, would you like to come on?
Yes, thank you very much, Prof. Schiestl. That was a really fascinating talk. I have 3 questions for you, if you don't mind.
Okay, sure.
So the first is, with the brassica experiments that you've done, has anyone thought about testing, what happens when you bring the divergent populations together again? So like, the populations that have evolved under bumblebee pollination versus syrphid pollination, and then mixing and matching, the different evolved phenotypes with different pollinators and seeing how that affects the evolutionary trajectory.
Yeah, we have done this. We have crossed the plants after the pollination. This is a very good point, of course, because it would kind of be the prerequisite for speciation, that some incipient isolation has evolved in here, but we have not found any, to be honest. So there was no effect of reproductive isolation in this experiment. So it would have been very nice to show it, but I think that it just needs more time, probably.
Yeah. Okay. So I mean, it wouldn't has anyone sort of tested whether it might result in character displacement or something like that, that kind of reinforces the differences?
Yeah, so you mean kind of a reinforcement scenario?
Yeah.
I guess you would expect this if you had some incipient isolation, for example, post-zygotic isolation leading to lower fitness of the hybrids.
Right.
Then you would have selection against hybridization, which could lead to reinforcement. This would be a beautiful experiment, but we have not carried this on because we haven't found any isolations or no post-zygotic effects of the crossing. So it would make little sense then to go on. But if you had this, then it would be nice. It would be the perfect setting for a reinforcement experiment, which could then lead to the fulfillment of all speciation criteria.
Okay, cool. Thank you. And then my second question. So I noticed that the evolution of scent compounds in the bumblebee plants, so the phenotype shift was like very rapid. The phenotype was pretty stable for the first up till I think the 9th generation or something, and then there was a crazy spike from the 9th to the 11th.
Right.
So why is that? Do you know?
Yeah, it's typical for experimental evolution that very rarely you have this nice sort of increase that you would expect for directional selection to cause. But it's just different effects that you have. There's of course one important fact is inbreeding. So we have quite a bit of inbreeding, and then in the end we cross the plants. Actually, so no, we cross the replicates of the treatment group. So we didn't cross the treatments, but we crossed the replicates within the treatments, which overcomes all the inbreeding effects. And then you actually see you see the evolutionary change without inbreeding. And this is one of the explanation. And then it's also, scent is a highly variable character environmentally, and slight environmental variation throughout the experiment. You cannot avoid this. Because the light, for example, in the season, the lighting is different. This is done in the greenhouse, and you still have natural light. And that light is different in winter, it's different from the summer, for example. And this has an effect. So this also contributes to this variation across the generations that is just unavoidable. No environment is perfectly the same all over the year. So.
Okay, cool. And then finally, the experiments with the spiders, I was wondering how the presence of spiders affects pollen transfer rates. Because you said the bees can kind of recognize when a spider is on a flower. So maybe that would lead to lower pollen transfer rates or something like that. And, it could affect, or it might not influence female fitness, but it could contribute to flower evolution through the male pathway.
Yeah, I agree. I totally agree. Yeah, that's especially, I mean, if you know Biscotella has just 2 ovules per flower. So there, the female function is quickly saturated, but the male function is more likely, more strongly affected. And I mean, lots of people argue that most of floral evolution is driven by the male function of the flower, which is a lot more variable and more subject to evolution, to selection, because there is more possibility for variation in this function. That's true for animals and plants. I mean, that's
cool. Thank you very much.
Yeah, I agree.
Thanks so much, Aaron. Ethan, would you like to go ahead?
Yes. Hi, Florian. Yeah, thanks for a very inspiring talk. So with the example of sympatric speciation between the 2 Ophrys orchids, I just wondered, what did the distribution ranges look like, and whether the site that you worked in could potentially be a contact site between the 2 species. So meaning that perhaps they've got very similar abiotic niches, and the site that you've worked in might just be a secondary contact site. I mean, just for interest. Just like
yeah, sure. I mean, this is again, I mean, this is generally the problem for making a strong case for symbiotic evolution in the field, that you don't know the history of the distribution of this of the plant or of the organisms in general. I think our case is one of the more stronger ones, because we have this, if you remember one of the first slide, I showed the Ophrys insectivora has this broad distribution all over Europe. And Ophrys ioannini is a tiny local endemic within this huge distribution in Europe. So it's maybe 1,000 times less distribution. And then this is on the big scale, on the geographic scale, but then on the microhabitat scale has broad overlap. So we have most of the populations that we have seen are broadly sympatric. I know there's a study that has also found some ecological segregation between the 2. But it's not really clear to me how they did that. Maybe they just selected the allopathic populations. But all I can say is that we have seen, we've also done maps, actually they're published as a supplementary to the one paper. We've done these maps, and we've seen plants usually growing side by side. So, and the pollinators easily fly, they fly 100 meters, no problem. So in terms of pollinator activity, this is a very clear, fully sympatric distribution. So I think, I know this is controversial, but I think our case is more strong in this respect.
Very interesting. Now, because here in South Africa, I mean, you often see it where a lot of these things that are pollinated by long proboscis flies, you get the same species, but then just different ecotypes, and you often find shorts going with longs. And they're also pollinated by different, or flies with different proboscis lengths. Often they co-occur in sympatry. So it's just something that kind of reflects, and it's very similar to the scenario that you see in the field. But yeah, interesting work. Yeah.
I mean, if I just add on this, I think there was one study by Peter Anderson, I think, that looked at disruptive selection in one of these, I can't remember the genus. I'm sure maybe Timo knows, or Ethan, maybe you know. It's an Amaryllidaceae, I think. And I think they found disruptive selection in one season, but not in the other. So it's
all right. I think that's Laperosia anceps. Yeah, it's earlier.
Yeah.
Bruce Anderson, yeah. It was my PhD.
Bruce Anderson, yeah, okay. Yeah,
yeah.
Sorry, I mixed it up a little bit.
Yeah.
Yeah. So there's some.
Thanks.
Yeah. Thanks, Ethan. Glynis, would you like to go ahead?
Yes, thanks, Sandy. Thanks, Florian. That was so interesting. Really fantastic, fabulous work you're doing. Thank you. My interest is actually in your future work that you're planning, where you're looking at soil type, and I wanted to know if you think that there would be changes, or perhaps there's evidence in the literature for changes in nectar or scent because of different soil types. Is that something that's known?
I would say there's very little data on this. I mean, what you've seen with different soil types, of course, if you plant the same genotype of plant in different soil types, you see massive plastic effects, because the amount of nutrients that change the architecture of the plant totally. But then if you look in the field, we see evolved changes. I think that's a different pair of shoes. But in terms of, I don't think there's much data. I mean, what we have seen in terms of evolved changes, we've looked at the Brassica incana population in southern Italy, and we have found that plants on limestone, these grow on limestone and on tuff soil, and limestone is a very bad soil, so to say. It has like this high concentration of carbonate ions, and the plants have massive troubles to get their nutrients. So on the poor soil, we have seen a high concentration of nectar and longer stamen, curiously. And that's over 20 populations that we have investigated. And also scent, there was a tendency to produce more scent compounds on the poor soils. So it seemed like those plants produce more attractive, or tended to have more attractive flowers on the poor soil as compared to the rich soil. But I don't think there's much in the literature about this in general. So this is a study we are preparing now for submission, so it's unpublished data.
Okay, well, thank you. Thanks very much. It was interesting.
Sure.
Thanks, Glynis. Timo.
Yes, thanks, Florian. I really enjoyed that. I was wondering whether you know the genetic background of the divergence. So I was particularly interested to see that in your Brassica experiment, the bumblebee replicates all sort of converge towards similar phenotypes. Do you think that, well, or even no, because I guess you still have the original generations, whether they are potentially different de novo mutations that happen in these three replicates, or do you think it's basically selection on existing genetic variation? And I guess in the latter case, you would expect that some of your original first generation individuals would already carry those genetic signatures that then are selected for in the various replicates.
Right. So so the questions about novel mutation, I think most likely the changes that we see come from standing variation in the population. So I don't think there's a lot of contribution by novel mutation, simply because we know mutation rate is very low. I mean, there's 1 or 2 de novo mutation in Arabidopsis per generation. So there's very few opportunities in such short time to create divergence by novel mutations. And then one thing one should not forget is that this is a full outcrosser, so we have recombination in each generation. So it's always a full set of new genotypes that are created in each generation. And recombination is an incredibly powerful process to create new variants in populations that then selection can act upon. So I think it's the factor of standing genetic variation and then ongoing recombination that causes these the divergent phenotypes. Did this answer your question?
Yeah, thanks. Yeah, that is based on the results that you showed. That seemed the most likely scenario, because the phenotypes become so similar that I guess that makes sense. Yeah. Cool. Thanks.
Yeah, you're welcome.
Hands up or questions in the chat. Just wonder. Cool. Then, yeah, it's 6:20. I think if there's no other questions, we'd like to just really thank Florian, and especially since you got up early in the morning to do this presentation with us. So thank you so much. And yeah, it was great. I learned a lot, and yeah, hopefully everybody yeah, I can take note of the recording after this is also, if they want to go back to it. I know Steve was asking for the recording as well, because he unfortunately had another meeting today. So yeah, but thank you so much. We really appreciate it. And hope to see you in South Africa again soon.
Yeah, hope I can make it at some point after Corona,
Yeah.
maybe.
And all the best with the rest of your sabbatical.
Yeah, thanks very much. Yeah.
And just to the SAAB community, just remember that if you need us to record your SACNAS points, please to put your full name in the chat as well. But yeah, thank you so much, Florian.
Yeah. Bye-bye to everybody.
Bye.
Bye.